Good morning, everyone. It's Sean from STK Markets here today. Today we've got senior management from Aegros, and I've got some terrific news, which we're really happy to let you know about. Hi, guys. How you going?
Very well, thank you, Sean.
This is an awesome day.
Hey, Sean.
Awesome day. One for the record books. We've got a few questions just to get some updates. We're going to finish really strong today. There's some wonderful news that we've got to give an update on. Guys, thanks for your time. What I'd like to do is just to get a quick update on Singapore, how that's progressing, an update on Queensland and your strategic investor. They're the three short topics that we want to cover quickly, and then we'll get to the really big news.
All right. Can I first start off by introducing Dr. Leon Rosen, who is our Chief Medical Officer. Leon joined us a few months ago, and he's very much been in the front and center of the clinical trial.
Hello, everyone. Nice to meet you virtually.
Yes. So let's first start. Thank you for organizing this, Sean. And it's always good to talk to our shareholders. As far as Queensland is concerned, we are well within what our targets are in terms of getting our development approval. We expect that development approval any day now. We've been told that it has gone through Ipswich Council, and I do know that some of our senior staff have meetings with Ipswich Council later on this week.
Yeah. So at this stage, we're still expecting to break ground by 31st of December.
Yeah.
In terms of the Singapore, we are well down the track. We've already communicated with the Singapore government in terms of the benefits that they may offer us. And I'm expecting again that we can have an update on that probably in about a month's time. Strategic investor, yes, that is progressing as expected. I do expect to conclude end of this year. It could push through to the beginning of next year. Fundamentally, Mac Bank is doing a great job. The strategic investors, which include a number of fractionators, have seen our facility. They are becoming comfortable with that. We have sent them some product which they will be testing in the next month so that they can see that it is exactly as we said it is. So I'm expecting that to progress to the next level because demand for these products is going up. The yield is a very big issue, and just the whole cost and the environmental advantage of our process make it really the only way forward. In addition to that, we're moving forward with our Australian commercial strategy, which will become the point of another discussion, I think, in our next meeting, but it is moving forward.
Okay, that's great. So everything appears to be on track there. Now, the main thing that we're here today for, which ahead of yourselves, we've obviously been focused on this clinical trial for quite some time. This is a pivotal point for Aegros. Like any biotech company, these trials are very important. It's make or break. What news do you have for us today, please?
Yeah, but first, before we start on this, yes, this is a very, very exciting day for us. And what we're going to say is actually report on some preliminary data. And I want to be very clear about it. This is preliminary data on our Sentinel group. And this is the group of 3 that we've done. And I'll hand it over to Leon in a minute for us to talk about this. In no way is this to be taken as the final medical scientific conclusions on the clinical trial. And so that we are also clear, this is in no way a promotion or an advertisement for any product that Aegros will be selling. With that, I'll turn this over to our Chief Medical Officer.
Thanks, Harry. Hi, Sean. So the study, as you probably remember, is long title is Intravenous Immunoglobulin Rich in Neutralizing Antibodies to SARS-CoV-2 as a Passive Immunity Modality. It is an open-label, active control phase 1/2 study. In terms of the primary objective, there's a number of primary objectives that we have. The one that I want to highlight for today is to really assess at day 42 the pharmacokinetic similarity of our product compared to convalescent plasma in healthy volunteers following a single intravenous infusion. Just to reiterate, pharmacokinetics is what happens to the drug within the body. And we know that with an intravenous infusion, 100% of the product will actually get into the bloodstream. And then over the next 42 days, we will actually monitor what happens to the levels of the product in the bloodstream. So the two products that we're comparing is our Hyperimmune immunoglobulin, which is being formulated to a 5% IgG, 32 mils, and equivalent to that in terms of antibody levels, equivalent to that is found in a 250 mil plus or minus 50 mil donation of plasma with a neutralizing antibody titer of greater than 1 in 160. 250 mils is approximately the average amount of plasma that's taken from a blood donation, and 1 in 160 is the titer that the FDA recommends that antibody level be above for it to be used as convalescent plasma in therapy. For the convalescent plasma, obviously, we are using that 250 mils plus or minus with the neutralizing antibody titer of greater than 1 in 160. So what we are looking for is that we get at least the same levels of antibody from our immune globulin that we would get from a single dose of convalescent plasma. Why are we doing this, and how did we come to decide that this was a product that would meet an unmet medical need? Well, the best protection against severe disease when you have a viral infection, especially a respiratory viral infection, is to have antibody responses at the time that you are infected. Now, that's the whole logic behind vaccination. However, there are some patients out there who cannot, for one reason or another, form antibody responses to the vaccine, and their response is either blunted or potentially non-existent, and particularly in individuals who have moderate to severe immunosuppression because of an underlying hematological malignancy like a leukemia, a myeloma or a lymphoma, or sometimes because of therapy for various different things that are immune suppressants. They will not respond well, and their risk of having severe disease if they're exposed to COVID is still there and still significant. So we do believe that there is a significant unmet medical need still out there, even though vaccines have got very good coverage. One of the potential advantages of the immune immunoglobulin over convalescent plasma or even a monoclonal antibody is that there will be a diversity of antibodies in there. And so we are more likely to be able to cover variants that are coming along with the hyperimmune than potentially with either a convalescent plasma, which is only from one donor, with vaccines, which the virus may actually develop, immune escape from, and the same with a monoclonal antibody. So what did the CHAT study actually look like? So as you know, we did 11 healthy volunteers and infused them with convalescent plasma, and that was completed. And then we were looking at doing healthy volunteers and immune-compromised patients, infusing them with hyperimmune globulin. And the first 3 healthy volunteers that we did were our Sentinel safety group, and that has been completed as well. Sentinel group were 3 healthy volunteers. They were infused with the product and they were monitored overnight in hospital for safety and tolerability with the product. The safety data was then reviewed by the safety committee, which included myself, it included the 2 principal investigators and an independent safety monitor who had total veto over any decisions the other 3 of us might make. And the recommendation to proceed after reviewing the data was given to the ethics committee, and they acknowledged the ethics committee, and we have now moved on to infuse more healthy volunteers. The early individual results for the Sentinel participants are what I'm going to be presenting today. Obviously, we don't have them out to day 42 at this point. And because they're very early results, we can't make any statistical observations or conclusions. So we'll start with what we saw with the convalescent plasma volunteers. This is a little bit of a colourful graph, but what you can see here is that at baseline or day -1, all of these healthy volunteers essentially had no antibodies. If you think about the timing of when these were done, this was prior to vaccines and prior to a lot of people in Australia having been infected with COVID. So it's no surprise that there were no circulating antibodies at that time. And you can see there that all of them had a rise in their antibody levels after the infusion. That rise was sustained for a little while, and then over the 42 days eventually waned, which is what we would expect to see. And just to make it a little simpler as we move forward, what I'm going to do is just present to you the average for those convalescent plasma receiving healthy volunteers. So that's the thick red line. At this point, I'm only going to compare up to day 2 with the data. And here we can see the levels that we got, the incremental levels with the healthy volunteers that received the immune globulin. This is incremental because at the time that we infused healthy volunteers with our product, they all had some level of antibodies. Obviously, they had been vaccinated and or infected at some point. So we're just looking at how much extra they got. The bars on the red line at the bottom show the range that we saw with the convalescent plasma healthy volunteers. So what you can see is that with the immune globulin, all 3 of these healthy volunteers got levels or ended up with levels much higher than any of the convalescent plasma receiving healthy volunteers. Just to show this in another format with a graph that may be easier for some people to see, we can see that our 3 healthy volunteers that got immune globulin had increases in their antibody levels of between about just over 400 to 800 units per mil. Whereas the healthy volunteers that got convalescent plasma, on average, received an increase of about 150 units per mil. The range of that increase was from 88 to about just under 250. So we can see that all 3 of our healthy volunteers that received immune globulin ended up with titers and increases in titers much higher than any of the volunteers that got convalescent plasma. And just to summarise that, we can see that certainly from the early shape of that PK graph or pharmacokinetics graph, the profile of immune globulin seems to be similar to that of the convalescent plasma, which provided a boost of about 150, as I said, AU per mil. The increase when we used our immune globulin was at least 3 to 5 times higher than what we saw with the convalescent plasma. And the safety that we saw with all of with the Sentinel group, and so far with the other healthy volunteers that we have infused, really is consistent with what we would expect from any intravenous immunoglobulin product.
So literally what we're talking about here, just to summarize what Leon is saying, is a product that has met expectations in terms of what we expect a hyperimmune to do when you provide that to a particular subject. We are currently doing more subjects. I think right now we can say that we've done up to 10 altogether. We're looking to complete this in the next couple of weeks with a few more volunteers, but the trial is going really well. Now, I think that the most important aspect of all this is the first time in the company's history that we have manufactured a protein, a hyperimmune, which has been then given to a human being, and it has been shown that this particular product that we've given to the human being is safe within the parameters of the 3 subjects that we've seen. So what this has shown is that within the parameters of the 3 subjects that we've had a look at, and also with the at least 10 that have been done altogether, the product seems safe. But obviously we will be doing further analysis, and by the time this particular trial is completed in the next couple of weeks, we should be able to provide everyone with a full report, and a report that will also look at some statistical analysis as well. Big day, really big day for for Aegros, and it is something that John and I are very proud of. Absolutely. Because after all these years, this is the pinnacle of actually showing the world that we are not there just as maybe this would work company. This is a company that is manufacturing a product that may be used in the future for at least passive immunity in subjects who need passive immunity when you have a pandemic like COVID.
Yeah, I mean, the assumption was always that as this is a new technique, a new way of purifying the product, that somehow it would produce a less than optimal product. And what this shows is that, on the contrary, it actually produces at least as good a product as any other hyperimmune produced, and with all of the advantages that we have previously talked to. But I think it's also time that we consider how we take this to the market, and that will be a discussion for another video, as today we are here purely to talk about the clinical trial results.
Yeah.
Okay, this is fantastic news. So this trial, this data has just proven that the antibodies increase, which is what we've always wanted to see. It's proven that it's safe. So the two key things that we're looking for have been executed in this trial. Is there any reason we only showed 2 days? Is that all the data we have at our disposal at the moment?
Yes, so because of the timing and the time that it takes to actually do the testing, report on the results and then analyse them and etc., we've just got the first 2 days. In terms of when we expect to see a clinical benefit, or when we expect the clinical benefit to actually happen, it's going to be in the first few days after you actually give the product. Because the patient will have been exposed to the virus at that time. They will then get their passive immunity. So that's the time that the antibodies will actually need to work. So from a clinical perspective, that gives us the confidence that if passive immunity works, we're providing a reasonable amount of passive immunity. In terms of taking it out to 42 days, and we're getting close, and I think we're probably just about to see the day 42 results for the Sentinel group. That will give us an inkling and an indication of how long that particular antibody hangs around in the body for. We know that the antibodies after vaccines and after infection tend to wane. We want to see how quickly they wane after we give them passively, so that in case that somebody does want to give a second dose, not that that's something that we are currently testing, but we will at least be able to provide the information of what the half-life is of the antibodies and when you can expect that there won't be any left, which will be useful for some of the clinicians in terms of managing those patients.
So now that we've seen the results, the antibodies do go up. It is looking fantastic. We've got that safety. The pathway to revenue now, is there a market there that we can still see?
Yeah, there is definitely a market out there, and it's a market based on people who cannot produce antibodies themselves, the immunocompromised group, people who are patients or cancer patients, people who've got cardiac events where, vaccines cannot necessarily provide the necessary immunity that they need. And Leon, do you want to talk about this trial that's being conducted?
Yeah, so there's certainly, I'm aware of at least one other study in Europe that is looking at immune globulin in the same circumstances that we are. And in the US, they have been using convalescent plasma quite liberally, and we have been talking to the leaders in that particular field in terms of moving forward with clinical studies as well. I can also say that I'm aware that in Australia, a study on convalescent plasma in immune-compromised patients in a slightly different situation to our patients that we're talking about, but certainly with COVID, looking at convalescent plasma in these patients as well. So there's certainly no shortage of people thinking about passive immunity. What we're hoping is that we will actually be able to provide a much better way of getting that passive immunity.
This is not a market that's going away. On the contrary, it is unfortunately going to be with us for the rest of our lives, I think.
Yes.
Okay, well, thank you so much for that update on the trial. That's just, I'm a big shareholder myself personally. I put a lot of money into this company myself, and to hear this information. I can't wait to ring my wife and tell her that we've made a good decision, because this is a monumental day that we've seen these results. Now, I won't keep you much longer, but I just want to go through the milestones of what we're trying to achieve over the next 6 months, and where we're going to go to from there. And then the current capital raise that we're doing, and an offer that we've got to existing $5 option note holders. So can you just guys give me the update on the milestones?
Sure. So in terms of this, now that we've proven that this product works, and there's more obviously to come out of that, but we are no longer talking about maybe something's going to happen. So we are now press the button to do the upgrade. And this will lift the capacity of our facility here, our Hyperimmune facility here, from 30,000 litres to 100,000 litres per annum, with a further 100,000 litres that will come through in basically 18 months' time. That's a $65 million investment that we'll be making between now and the 31st of March. The two dates that we all need to think about are 31st of March for the approval by the TGA of our facility as a good manufacturing practice facility. That means we're making a product reproducibly in compliance with what the standard operating procedures say. Okay, so the second aspect is the ARTG registration, the Australian Therapeutic Goods Register, and that is the register upon which our product has to be inserted so that we can then sell a product. That's again with the TGA. And these are...
Yeah, and we've actually already started putting a dossier together, and that dossier will go in at about the same time. So that is literally something that has to happen in parallel with the GMP certification of our facility. And by the way, the upgrade is something that we should all be proud about, because this is the state-of-the-art hyperimmune manufacturing facility. And, you know, it comes with some equipment that many fractionators around the world do not have. So we really are doing something really special here.
It's going to be leading edge. And the important thing is, again, single step versus multiple steps to get your product, which means you have much higher yields, which means you have much lower cost, and it means much less time. So all of those factors come in and make this plant. The day of importance is the 31st of March 2024. That is the day we expect to have ARTG registration and the TGA approval. From that point on, we can talk sales.
That's just wonderful. So I think, obviously, just touching the strategic investor for a second, these results, have you shared these with Macquarie Bank to share with strategic investors?
No, we haven't. This is something that is ongoing. The clinical trial results are something we've got to be very careful about. This is the first time we've actually mentioned the clinical trial results to anyone. Leon and his team will be working and putting together a paper, which is normal with the actual results. But once we get all the results together and done some analysis, as far as the strategic investor is concerned, we will talk about this. You know, there are times when you can actually, you're required to show them this. Right now, they do know that we are in the thick of a clinical trial, and it'll be part and parcel of our negotiations as we go on further with them.
Well, this certainly won't help, hinder those negotiations. This will certainly make it work so much better.
Sean, I think the question people have asked a number of times, you know, are you guys real? You can't get more real than actually manufacturing a product and then putting it into a human being. So
and getting the result you expect.
Exactly.
Yeah, that's wonderful. So just touching on that funding requirement for the upgrade, that's a serious amount of money. I know we've got quite a lot of money coming in from the $5 options. I did see a note from yourselves that we have got an offer which will articulate in the email body. If you have more than $400,000 worth of $5 options, if you exercise by the 31st of October, you get one $15 option for every $5 option that you exercise. If you've got less than that, you get one option for every two options that you exercise. And bearing in mind that we are raising at $18, we are getting money in the bank at $18. The cap raise is going quite well. That's at a discount to that $18 price, and those $15 options are actually worth $9. So there's a tremendous amount of value for exercising early to spend $5 to get an $18 share plus to get a $15 option. And there's a lot of value there for investors. And again, if you don't have shares or options, the raising price is at 18 at the moment, and we are looking for 10 million at that level. We have filled a substantial amount of that money. I've been in Asia quite a few times. There's a lot of interest from Southeast Asia. After the Singapore transaction went through, there are a lot of funds and private equity and high net worth individuals that are wanting to come through. We'll have more on that in the future, but the funding is looking quite good in terms of getting the cash in. So I just want to say thank you guys for your time. Is there anything else before we jump off?
No, just we'll be back on the same bat channel and update you on further news and strategy.
A great day for Aegros to be in Agrossian. It's those antibody levels are fantastic. Congratulations again.
Thank you very much.
Thank you.